Okay, in this video tutorial we're going to look quickly at transcription. So we're going to talk about what transcription is. So let's just kick off where we left off. So just to come back to this idea, we're looking basically at what does DNA actually do. So we've looked at the structure of DNA, but we're going to look a little bit more at the mechanism of how DNA actually carries out its function. So DNA is essentially is sometimes referred to as the code of life. And essentially what that means is DNA contains the recipes which code for proteins. And proteins are the moving working parts of a cell. So it's these recipes that code for the proteins. The proteins give our cells their function. It's the function of our cells that make us who we are. So ultimately it is these recipes that really are the recipes for our characteristics, what we look like. So these within the DNA, these recipes are called genes. So you can think of the DNA or the genome as the recipe book, and the genes within the DNA are the individual recipes. So a gene is simply a coding sequence of DNA, so a specific sequence of A's, T's, C's, and G's that codes for a protein, and that carries out some sort of function and gives us our characteristics. So DNA also has non-coding regions. These are often referred to as junk DNA. However, it's worth just noting there that essentially, even though it doesn't code for protein, it may still have other functions. So in terms of, there are some sequences that essentially regulate the expression of genes, or maybe just act as a little flag or a sequence that indicates, hey, there's a gene here, next to it. This is a place to start. Some of the sequences may have other functions, even simply an evolutionary function in terms of it may be empty space where genes could develop, or there are some areas where there may have been a gene which is no longer expressed, and so it's kind of a gene that's no longer used. So the junk DNA, that term is a little bit misleading, and we're starting to realize that some of those sequences may still have some function. However, there's a lot of it that probably has no function. Another function maybe even just in terms of organizing the DNA in the nucleus. There's evidence now that the DNA is reasonably organized within the nucleus. It's not just spread out randomly. So junk DNA is probably not quite a fair term either. But anyway, it's the genes that most people are really interested in, because those are the actual recipes for various proteins and characteristics within the body. So, gene expression. We can't talk about gene expression without looking at essentially the central dogma. So gene expression is the term that's usually used to when we're talking about basically what genes do. And gene expression follows a couple of simple steps, and this is called the central dogma of molecular biology, because it's basically so fundamental to molecular biology. And central dogma, it's kind of means it's the central truth or the foundation. Now we know these days there are actually a few, some small exceptions to this or variations, but largely this is the case. And as far as we're concerned at this stage, this is what always happens. So DNA contains genes which are transcribed into RNA. Now I want you to think of RNA. RNA is actually a very similar molecule to DNA. I want you to think of it as a temporary copy. So this is the recipe within the recipe book, and that recipe book isn't going to leave the nucleus. So we take a temporary copy, almost a photocopy, of just a single recipe, which will leave the nucleus in the form of RNA, this temporary copy, and then that is translated. Now translation is kind of the actual baking. So if this is the recipe, translation is when this recipe is actually used to bake the cake, or in this case, to make a protein, to synthesize a protein, and it's this protein that then carries out that function. So the central dogma is really that DNA is transcribed into RNA, and RNA is translated and the product of that forms a protein, and we'll look a little at that in a little bit more detail. So in this video, we're going to focus on this first step, translation, and look at some of the differences between DNA and RNA, and in a separate video we'll look at translation. But just to give you an overview here of the overall process. So yeah, the central dogma is kind of answering that question: how do we get from code to characteristic? And as part of the central dogma, originally it was taught that one gene produces one RNA, and one RNA gives you one protein. Now, not one molecule of that, but one type of protein. This is a recipe. Gives one gene gives us one RNA gives us one type of protein. We might get several copies of it. Now, that's not entirely true. There are some exceptions to that. So, you may still, if you got a question in an exam, you could still answer it according to that. At high school level, that's often still taught, that, one gene, one RNA, one protein. However, there are exceptions to that. And for instance, in bacteria, that's definitely not the case. Often an RNA actually codes for several proteins. But even in humans and things, there are exceptions. It's also often taught that essentially once this process is started, it can't kind of be stopped. And that sort of largely is true, and it makes sense, because why go through the trouble of transcribing the RNA and then stopping the process? Why not just stop it from the start, from the beginning? However, we're starting to realize that there are some mechanisms that control expression at the translational level. So the RNA is made, and then something interferes or stops it from being translated. It's much more rare. It's not as common because it doesn't make as much sense in terms of energy. It's less energy efficient. If you don't want the protein, you might as well stop it at the DNA level, rather than, why bother doing this first step if you're not going to follow it through anyway. But again, that's something that's often been taught, that once something's transcribed, it will be translated. We know these days that's not actually true, and currently there's actually a lot of research going into control at the translational level. I guess a lot of research has already been done at control at the transcriptional level, so whether it's transcribed or not. And a lot of even people at universities still sort of operate under the assumption that if it's transcribed, it will go through the translation process, etc., and you will end up with the final protein, whereas we know these days that's not always true. You can't necessarily make that assumption. But anyway, back to basics. Really what we're looking at here is gene expression is all about how do we get from our code, our DNA, or the genes within the DNA, to our characteristic. And the answer is that the DNA is first transcribed, we get a temporary RNA copy, then that's translated into a protein, and that protein carries out some sort of function. So let's have a look at this process. So firstly, transcription is carried out by an enzyme, and in this diagram we've tried to show this enzyme here. We've made it clear. This diagram is portrayed the enzyme as being transparent, so you can see what's happening inside it. So the enzyme is this kind of triangular sort of shaped bubble, is an enzyme, and this enzyme is called RNA polymerase. So let's just look at what that name really means. So RNA, you may recognize the first part of the name here, polymer. So if you've studied chemistry, you probably know what a polymer is. And then it's got this ending "ase". So "ase" is a common ending for any kind of enzyme. So if it ends in "ase", the name of something, in biology it probably means it's an enzyme. So a polymer is a long chain, a molecule that's made up out of monomers, single units that have been pieced together. So this enzyme is really the RNA piecing together, all right, making into a polymer enzyme. So it's the RNA piecing together enzyme. So it is really, the name tells you what it does already. So polymer, a long chain, or has been assembled out of small parts. So it's the RNA piecing together enzyme. And essentially what this does is it splits open the DNA and it reads off the sequence on one strand and uses nucleotides. But these nucleotides are a little bit different. and we'll look at how they're different in a minute. They're just very slightly different, but incredibly similar to the DNA ones. They've still got a sugar, a phosphate, and a base. It's just the chemical structure of the sugar is just a touch different. So we've got nucleotides, which are assembled on the strand of DNA to basically make a single-stranded temporary copy. So you've got the DNA is 2 strands, double-stranded like this with the bases in the middle, and a section where there's a gene will be split open so that the bases are exposed. And then the RNA polymerase will come in there and will essentially make a temporary copy by pairing up, matching up those bases. And so this will be a temporary single-stranded copy that can then leave the cell. So sorry for my terrible drawing there. I've got a slightly better diagram I can show you in a minute. So this process is called transcription. And if you think about the term to transcribe, if I said if I said I had my secretary transcribe this letter, what would that mean? Well, a lot of people would say that basically means to, you got her to type it up or to write it out. And that's really what we're talking about here. If you think of the old days, people had scribes. Scribes were just someone who wrote down what you said. So royalty and stuff, and people like that, often, they wouldn't write things themselves. They'd have a scribe who would write down what they said for them. And so here, essentially, it's the same idea. You're making a copy, but it's still in the same language. So it is really just making a temporary copy in the same language, transcribing something. And so this process is called transcription. The RNA can then leave the nucleus. So this is all happening within the nucleus. And so we're going to have a little bit of a look at that. So I've got a bunch of cells here, and if these cells are living, they'll be carrying out some transcription, transcribing certain genes. So if we zoom in, we are really looking in here in the nucleus. All right, so I'll just go back out again. You can see here the black dots here are the nuclei of these are onion cells. And if we zoom in there, you can see this is supposed to be, and in purple here now, is supposed to be the nucleus. So here you've got your RNA polymerase, your RNA piecing together enzyme. It makes a temporary RNA copy from by opening up the DNA and just a temporary copy of just part of the DNA. So this would be a gene here. We're making a temporary copy of a gene here. And that RNA polymerase can then snake its way out of the nucleus and into the cytoplasm. So this whole process is basically about making a temporary copy of a gene that can leave the nucleus. So before we finish up for transcription, we just need to look at briefly just the differences between RNA and DNA. So I've got DNA on the left here and RNA on the right. So first key thing, which is really quite apparent, is DNA is double-stranded. It's got 2 strands. RNA is single-stranded, and hopefully in the picture here you can kind of see what we mean by that. DNA contains the bases A, T, C, and G. Adenine, thymine, cytosine, and guanine. RNA contains the bases A, C, G as well, but instead of T, so instead of thymine, that's the one key difference here, instead of the base thymine, so remember bases are the steps of the ladder, we have this base here called uracil, a U. And if you look at the structure, it's actually very similar, except that, and I'll zoom in a little bit there for you. Get rid of my little circles. So if you see here, I don't know if hopefully that's clearer. If you see here on the thymine, so we're looking at the thymine here and the uracil here, this has a methyl group, a CH3 group here, and uracil just has a hydrogen here. So it's really only a very slight difference in the structure there, and you don't need to know it in that much detail, but the U and the T are actually very similar in structure. And the truth be told, now my understanding of this, don't quote me maybe on this one, but I believe that when DNA is made, these T's are actually initially laid down as U's, and then a methyl group is added to turn them into T's. So a T is kind of just like the mature, the final form of a U. Now, because RNA is just a temporary version of DNA, because it's a much more temporary version, they just run with the U's rather than adding that methyl group and turning it into a T. So biochemically, all T's actually start out as U's. So a few other differences. Sorry, let's just get back to there. Is that DNA is only found in the nucleus. So DNA is the entire recipe book. It's much too large to kind of have all that DNA leave the nucleus. Whereas RNA can move into the cytoplasm. So RNA is technically found within the nucleus and the cytoplasm. You also find some RNA in some organelles. So for instance, like the ribosome. Every set of 3 bases on the DNA is called a triplet, and 3 bases on RNA, a set of 3 bases, is called a codon. And you probably don't need to worry too much about that now. That becomes quite important when we look at translation, the next step though. So we just have a name for a set of 3 bases. So if we were reading down the sequence of this DNA, so if it was A, A, T, C, G, the first 3 A, A, T, that would be a triplet. The next 3 would be a triplet, and so we put them into little groups of 3. And in the RNA, those groups of 3 are called codons. Lastly, there's actually one more, but another difference, DNA is much more stable, which means it has a much longer half-life, and that makes sense if you think about, it basically means it lasts longer, and that's important because DNA, needs to be there for the entire life of the cell. And in fact, actually beyond that is actually passed on to the next generation or to the next cell. So the DNA, it's much more stable, which helps to ensure that it's not damaged or degraded, because that would have bad, have negative consequences. It's kind of our permanent copy. Whereas RNA is by comparison fairly unstable. So it usually only lasts a matter of hours, maybe a day at most. It depends a little bit on the sequence itself and a few other factors, but RNA doesn't last nearly as long. And that makes sense because if we want to make a protein, if the cell is going to produce a protein, it could express the RNA. So we could make a temporary copy of the RNA. But by doing so, we don't want to make that protein forever from then on out. So eventually the RNA is broken down because it's unstable, or enzymes come along and break it down. And then if you want more of that protein, you've got to make another temporary copy. So it's that level of control too. And so I guess that's one reason why these days sometimes it's controlled at the level of translation. The next level is basically that gives you a slightly quicker control or better control in terms of that timing. So you don't have to wait for the RNA to kind of be degraded and disappear. You can turn it off a little bit quicker, or on, in fact. But generally, if you've made RNA, you will then follow through to the next process. One other key difference that I haven't actually put in my little box at the top here, DNA is much longer. And if you think about that, DNA is our recipe book. So if you think about a recipe book, it's got a whole lot more pages, then RNA is just a copy of a single recipe. So it's a much shorter, the sequence of bases is a lot shorter than our entire DNA molecule, a heck of a lot shorter. So that's basically it for transcription. So I hope you found that helpful. I've got a little animated video that kind of shows a little animation of transcription that I'll chuck in the playlist too. So that'll be the next video, and then we'll get on with translation.